So good morning to everyone. So I welcome you all in my next lecture, that is BT technology and classifications. So in this class we will discuss about the BT technology, what is the BT, why this technology is important for the development of crop and different crop plants. So what are the different BT proteins are available with us to develop different class of new plant varieties, different class of then BT proteins. So basically, if we talk about the BT, in that BT generally stand for the Bacillus thuringiensis, that is a soil bacteria. So it is first isolated in the Germany. Okay. So BT Bacillus thuringiensis is soil bacterium gram-negative bacteria. This bacteria has a unique property, which produce a protein that paralyze the different class of insect larvae which are harmful to the different crop plant, which are harmful to our different crop plant species. That also include the cotton, tomato, and you can say our different BT brinjals also. So in case of urea and European corn borer also, they have common plant pest, which infestations produce large of devastating economic losses for the important different crops. So basically, if we talk about this introduction about this technology, the BT is this BT bacteria is easily cultured by the process of fermentations. This overall last 50 years, if we talk about the BT, that has an insecticidal by the insecticide effect by the different farmers in the worldwide. Organic farming particularly has benefited from the BT insecticide as it one of the most few pesticide which is permitted by the organic standards. This insecticide is applied either as a spray form or ground application. So we can apply this BT insecticidal formulations as either through the spray form or ground applications with the irrigations. So it is available in both form in markets like granules form and liquid form. If we talk about the efficiency of this insecticide proteins, efficiency of both the applications, either spray form or ground applications, is quite limited. So, as target organisms often do not come in contact with the insecticide, as they are found on the underside of the leaves and have already been penetrated into the plant species. So that is the main drawback of this technology. So, during application, not this technology, application, because of applications. So nowadays, this BT technology have been make some improvement in that. The scientists have taken the BT genes from the bacteria. Okay, so that is encoding the different insecticidal protein. Okay, so that gene has to isolate it from the bacteria and insert into the different crop plants. So these genes make plant as an they develop inbuilt resistance into the plants by incorporating the gene into the genome of the plant species, genome of the plants. Thus, these plants have a built-in mechanism to protect against the target pest. Protect against the target pest. Okay. So similarly, nowadays the scientists are working on that to develop the different insect resistance crop plant species. The protein which are produced by the plants does not get washed away or not get destroyed by the sunlight. So plant is thus protected from bollworm of on-borer round the clock regardless situations. Okay. So this is about introduction of the BT technologies. So BT, as I told you, Bacillus thuringiensis is a gram-positive bacteria, which is aerobic and rod-shaped. You can see in the picture, it's a rod-shaped bacteria. It's a ubiquitously is found everywhere, so there is no difficult to get isolate the culture of this bacteria. It form the endosperms. It form the endospores. Have a genome. If we talk about the genome size of this bacteria, genome size is ranges 2.4 to 5.7 millions. 2.4 to 5.7 millions of base pair. Okay. So it produces one or more different insecticidal crystals protein. It produces one or more different insecticidal proteins which occupy more than 90% of the world biopesticides marker. So this is one most advantage that is cover largest of the market nowadays. It cover more than 90% of the world market. We talk about the history of this BT, which is isolated in 1901 by the Japanese scientist, that is Ishiwara. Shigeten Ishiwara. So he discovered this BT bacteria. Okay. he discovered. So that is a very common bacteria which is found in different environment also, soil and desert, as I told you, first isolated by the Ishivata. Then in 1911, German scientist Ernest Berlin, again he re-isolated this BT from four different moth caterpillar that had been collected from the Thuringia, that is a different country one, in the Germany. Soon this Berliner that determined the Bt bacteria has a specific toxic protein that Berlin has concluded. This Berlin scientist has concluded that Bt has a specific toxic proteins against the certain insect and pest larvae. Okay. So notable, it was not until 1928 that anyone attempted to harness the Bt as a tool for the pest control. Earlier, no one is using. If we talk about the timeline of the discovery of this, in 1901, as I told you, Bacillus thuringiensis was isolated by the Ishivata in 1901. But he was not able to explain the insecticide and he just identified it. Okay. 1911, again re-isolated from the four moth caterpillars. 1928, BT as a used good tool for the pest control. First time it was used in 1928 as a tool for the pest control. First time commercial Bacillus thuringiensis based biopesticide introduced into the plant, which is widely used as a biopesticides. In 1990s, thousands of Bacillus thuringiensis strains has been identified across the globe. Okay. In 1995, the first genetic engineering corn engineered with the genes. 1990, first transgenic or genetically engineered corn or maize were engineered with the gene from BT, which become available for the marketing. So you can see the timeline in 1901 to 1955, 1995. It takes nearly 100 years to harness the capacity of this BT as a pest control or transfer into the gene into pest control. So it's a long history, about near 900 years. So this bacteria was used to fed off the European corn borer, Ostinia nubilis. That's a European corn borer. That is the most devastating pest in case of European countries, which historically have been a common and very damaging corn pest. Have been very damaging corn pest in case of Europe. This initiated the development of first commercial BT-based biopesticides against the European corn borer, Ostrinia nubilis, which is the most devastating pest in European countries. Then the initial development of first commercial BT-based biopesticides was sporin, which was introduced in 1938 in the France. Then the BT-based biopesticide has been significantly pest control strategies nowadays. Then no one has thought about the GM corn engineered with gene BT available in 1995. This gene have come dominate the majority of the varieties which is planted in the US, representing 81% of the total corn was transgenic with this gene, and 84% of total cotton acreages was covered by this gene. And if we talk about the cryproteins are toxic to the insect order Lepidoptera, Coleoptera, and Diptera, and also to nematodes, also to different class of the nematodes. If we talk about the size of this protein, cryprotein, these cryproteins are the resultant encoded proteins by the crygenes. Crygenes are nothing but these genes are present, Bt genes. The other name is crygenes. Bt genes are present in the genome of Bt bacteria in Bacillus thuringiensis. Okay. Then, if we talk this protein encode a protein product that is known as crystal protein. That is known as crystal protein. This crystal protein are also known as Bt crygenes, cryproteins, because they are encoded by the crygenes. Crygenes are present in the genome of the Bacillus thuringiensis. Okay. Then the size, if we talk about the size of this cryprotein, it is varies from 130 to 140 kilodalton. 130 to 140 kilodalton. The protein was characterized by the using the different HDS-based protocols. More than 500 different crygenes and they have been divided into more than 67 families, cry1 to cry67. Based on their property, based on their sequence analysis, based on their physical interaction with the insect insect and pest gut. They have been classified into different families. If we talk about the cryprotein structures, the N-terminal domain, if we talk about this structure of this cryprotein, The N-terminal domain is a bundle of 7 alpha helices. You can see the N-terminal domain, this is C-terminal domain 3, this is N-terminal. First N-terminal domain is a consists of 7 alpha helices. Domain 2 consists of 3 antiparallel beta sheets with the exposed loop region. Domain 3 is a beta sandwich. Now we use Gram-Stenick technique as it a gram-positive bacteria absorb crystal violet strain. It absorb crystal violet strains and appear violet in the colors. Endospore staining, endospore are stable resting stages. A vegetative cells normally transform itself into the endospore under unfavorable conditions. This process is called as sporulation. This process is called as sporulation. In vegetative cells will normally transform itself. Vegetative cell normally transform into an endospore under unfavorable climatic conditions. The process is called as sporulations. This process is called as sporulations. Bt crystals. They are also known as insecticidal crystal protein. All are proteins crystal formed during the sporulation in some Bt strands. Bt produce protein that aggregate to form a crystals. They form aggregate and form the crystals. If we talk about the different shape of these crystal, Bt crystals protein or insecticidal crystals protein, they have different shape according to their insecticidal activity. So if first is bipyramidal, if you talk about the bipyramidal shape is also there. That bipyramidal shape of cryproteins are ranges 130 to 138 kilodalton, 130 to 138 kilodalton of the proteins. If we talk about insect activities, Lepidopteran larvae. Cuboidal is 69 to 71. 69 to 71. Lepidoptera and Diptera. Lepidoptera and Diptera. Is flat irregular 73 to 74 Coleoptera. 73 to 74 Coleoptera. Bipyramidal 73 to 134 Diptera. 73 to 134 Diptera. Now, various step or other various protein which are ranges 35 to 129, and there are various of different insecticidal activity. You can see here the different shape of the crystal proteins, bipyramidal shaped crystal, spherical shaped crystals, and rhomboidal shape also you can see here. Cuboidal shape is also there. Okay, these are the structural differences between the different shapes of the proteins. So this picture you can see. Now we take the preliminary test and how we can confirm that and characterize the proteins. So based on the colony color you can result to be obtained that is creamy color. If we are taking colony colors, shape of bacteria, rod shape, gram-stenic because they give you gram positive. Okay, so these are the features of the bacteria based on this tech. Spore staining showing that the presence of spores is there or not. Crystal staining, they are showing the different shapes like cuboidal, pyramidines, bipyramidals. Motility test, motile. Motility test is motile or not. Shape of spore showing ellipsoids. Shape of spore ellipsoids. Location of spore is showing that it's central. Length of micrometer is 3.13 to 4.75 varies. Width is 0.85 to 1.04. 1.04. Biochemical test for their classification, they have biochemical physical type of tests like Turin-Genesis, Kushaki, Indent, Gallery, Alto, and Dendrolimus, Morizori, Dermastanders, Austrinia, Israelensis. These are showing different biochemical